It's a pleasure to be here. I think we're entering a new way in which we're going to use data to improve the survivorship of diseases like cancer. And I'm very excited to be able to talk a little bit about it tonight. So I think most of us can think back to a time that we learned that one of our colleagues or one of our friends had cancer. And so generally there are two questions that come to mind. One is what is the survival, how long will they survive? What drugs will they take? And are there some better drugs that might improve their survival? The second question is, people tend to feel a little futile when they ask this, but the second question they ask, is there anything that I can do that might help my friend or colleague with cancer? And those are the two sort of questions I want to have you think about over the next few minutes. So in front of you are 6 slides, histology slides of two types of cancer. One is, they're both lung cancer. One is lung adenocarcinoma. One is squamous cell carcinoma. And for many years, we use instruments like microscopes to decide, what type of cancer you had and what the treatment would be and what drug. And this is sort of in the analog age. And there was a certain, as we heard at the beginning by Mark Retain, accuracy and precision are difficult sometimes with data like this. So I don't know if you could read all of these, so I'll give you the high-end view. These are a lot of G, A, C's, and T's. This is sort of the genomic basis for not only the cells, but a lot of the other processes. And as a computer scientist, and Mark, thank you for, I am a computer scientist, so don't listen very much to me, but the one thing I know as a computer scientist is a lot of what we do with computers and information begins with an alphabet. That alphabet is usually 0 and 1, and from, the absolutely fascinating thing about biology and how we use biology to look at disease is this sort of miracle from a sort of an intellectual point of view, which is very, very deep at some level, that we code up our entire body and we code up a lot of diseases using a 4-letter alphabet. And it's a little more complicated than that, but that means some of the basic tools we've used can be used to look at biology and disease and how to treat that. And from the viewpoint of someone like me who came to this field relatively recently, we by and large don't use any of the digital tools that are available that we can apply in this area. And that's the opportunity, and that's what I want to talk about. So the first thing you could do is you could take these analog slides and you can sample the tissue and you can sequence that, and then you can look at the genes that are mutated, and you could begin to decorate. We use the word annotate, so we seem more grown up when we talk about that. But you can annotate these slides with the genes that are mutated. Let's assume these are single, a single base that you saw on there was mutated. We could begin to annotate those. And that's the first thing we did. And for many years, the people in this field took the view that if we understood the genes and we understood the mutations, that was enough to give us the insights we needed to treat many diseases. And unfortunately, most diseases are not that complicated, or not that simple. Most diseases have rich combinations, and that has two issues. The first issue is how do we understand it? The second issue is how do we sort of gather the data and analyze the data, and then how do we action? And I want to talk through that. So unfortunately, something as simple as looking for the right genes and the right mutations just doesn't give us the power we need to attack this. The other thing about when we looked at it this way, when the field was beginning, we took probably a few dozen patients and we tried to see the patterns there. And if it really were as simple as what is the mutation and what do we do about that mutation? You know, dozens to hundreds of patients would have had the statistical power to give us what we need. Again, we were disappointed. So the next major step was a project called the Cancer Genome Atlas. And this was given birth in a period in which the National Cancer Institute had funding from the ERA project. And you know, approximately $400 million was spent to sequence and gather data about initially 500 patients for 20 diseases. So that's about 10,000. We're up to about 12 to 14,000 patients now. And what we did is we went from a gene to the whole genome. And this is the observation that, you know, if we look at the whole genome, the genes are only about 2% of the information. And again, look at the alphabet and the information coded. From a pure information viewpoint, there were two choices. Choice number one is nature is wasteful and doesn't really know what they're doing. Evolution is wasteful and doesn't know what they're doing. And they took this massive genetic information and say, oh, 98% of it's junk, we don't care, it just came there, and 2% is relevant. And you know, that's what failed, that approach. And the approach of the Cancer Genome Atlas, and it's a very successful project, was to look at the whole genome, look at not just the DNA, but the RNA, look at all the different kinds of measurements you can take and try to understand it. And again, we went from the gene to the whole genome. Again, diseases like cancer are just very, very complicated. And here's a simple example. In this type of lung cancer, for a particular tumor, if we sequence the tumor, the average number of mutations is 360. The average number of rearrangements, if I take a little DNA here and take a little DNA here and rearrange them, sometimes fuse them, the average number of rearrangements for a tumor is 165. The average number of what are called copy number variations, where a significant overexpression of a protein is present because you take a little piece of that DNA code and you replicate it multiple times, so you have multiple copies. In that, there are over 320 copy number changes. So not all diseases, or not all cancers is complicated, but the whole genome approach also failed. Now, on the other hand, there's an incredibly rich amount of information here, but this is a problem that involves a lot of data. And so, you know, our group over the last several years has built a system to analyze whole genome data at the scale that was collected for Cancer Genome Atlas, roughly 10 to 15,000 patients, and at the scale it's going to be collected over the next several years, roughly 100,000 patients. And data at that scale has not been consistently analyzed up to now, except by a handful of groups in a handful of cases. So there's an incredibly rich amount of data, some of which will be actionable, that we can get from here. And that is, this is going to launch in a few weeks, and we're going to, if you come after this talk, you can see a preview of this project over there, and you can see it, what's actually going to be available to any researcher. And I think this is the beginning of what you might think of a third era. And I want to describe the third era right now. You could think of this as going from the whole genome to a whole data approach. And so this is what a commons is about. And so I'm going to describe it this way. So I think of a commons like the one we built for the Genomic Commons as like a big food court. And imagine if you have to go home after this lecture and you have 6 children. Now, does anyone here have 6 children? Okay, but imagine you have 6 children. And you have to feed those children, and each of those children, because you came from here, each of those children, you're going to go out. But the problem is each of those children wants something else. So one approach is for you to go home, find out what the children wants, and say one person wants McDonald's, you go to McDonald's, you get the McDonald's, and you bring back McDonald's to home. And then you find out the other child wants, a burrito. So you go out and you go get the burrito and you bring it back. And with 6 children, at least with the kind of children I know, you won't be finished bringing back all the food and doing the orders till well past their bedtime, almost probably the next morning. And so, let's say you want to have a tasting and you want to taste ice creams. So you want to go out and each child gets a different brand of ice cream, Haagen-Dazs and some of the fancy gelatos and so on. And you bring those back one after another. Again, to taste all the ice creams, all the ice cream is going to be melted before you can taste them. So the idea of a commons is the same idea as a food court. You get a big mall, sort of an industrial-sized mall, and you put the food court in, and then you take your family here, and they sit here, and then you just sort of walk and get the food and bring it back. It doesn't take very long. You sort of jointly eat the food. If you want to taste testing, you sort of just get that and you do it. And it sounds like a simple idea, but we didn't have malls for a long time, and it's the same way we didn't have commons for a long time. And so we've built a large-scale commons so you could bring all the cancer data in the world in one place, and so the oncologists and the researchers don't have to make multiple trips. They don't have to set up their own infrastructure. They could just come to the mall or the commons, analyze the data, quickly instead of taste tests of ice cream, they could quickly apply different informatics tools. Anything they could want is somewhere around there. So, and you could also have not just one type of ice cream, but multiple types. So the way we think of that is it's no longer whole genome, it's genome. It's all the clinical data. It's any environmental data we can get. It's any pharmacological response we could get. And so it's a different era and a different scale. And the only reason it works is because companies that care a lot about you, like the large advertising companies, have created very large-scale infrastructure and large-scale cloud computing to give you better advertising. And we can borrow from those ideas to sort of manage the data and analyze the data at the scale required to analyze whole genome, all the clinical data, the environmental data, and everything else. And so that's good planning. And so if we go back to those 6 slides, and I don't want your night to be spoiled, so I'm going to tell you which was which. The top 3 were adenocarcinoma, and the bottom 3 were squamous cell carcinoma. But it's quite hard to tell from the genes. It's a little harder to tell from the whole genome. But if you take all the data that was collected by the Cancer Genome Atlas, there are about 300 cases if we pool those things together. And you could do a mathematical analysis because all the data is in a commons, and we do the mathematical analysis, and you could do a technique called principal components, and we get a clean separation between the gray adenocarcinoma and the purple squamous cell carcinoma. And we see that there's a simple hyperplane that more or less separates these things, but the green seem to be misdiagnosed. So things that when we looked analog and we looked with the current single gene and so the technology until recently were quite hard are much simpler with this level of data. So I think one of the lessons if you work with large data is many problems have become much simpler with scale. As long as you can manage the data and analyze it and keep your wits about you, then things become simpler, as you see in this example. And so if you think of each tumor that you sequence as having a genomic signature, and we sort of, here I've written one drug, but typically there are multiple drugs, we record all the drugs, we record the survival, then at scale, with enough data, you could do something pretty simple. You could take a tumor from a patient, you could sequence it, you can find which is the closest signature, you could find which drugs for that signature for that patient with matching clinical information has the longest survival. So you get a ranking. And this is a paradigm that's very common in computational advertising. It's an action-driven paradigm, and at scale, it could be used here. And so there's no standard term for this. So the way I like to think of this is you combine traditional bottom-up, hypothesis-driven root cause analysis, mechanistic view, and in the end, if you understand how cancer dysregulates the networks, understand how to target those networks, you can eventually get a drug that's targetable. But you can also come top-down. With all the data, you can sort of collect the data, categorize data, analyze the data, rank the data, and as I just showed, take an action. And bringing together both of these in a commons is a very powerful approach. And we're launching this, and one of the reasons we're launching this in a couple weeks, and we're previewing it tonight. One of the reasons I'm excited about being here is we're already thinking about how to design a system for 100,000 patients, how to make it not just for an institute, but how to make it for a consortium of people, and how to sort of go back to that question I began with. I already talked about one of them, which drug or combination of drugs. Just look at the data and go top down. The second question, what you could do, I want to end with, we're designing this system. It's a commons that will be shared across multiple medical research centers. It's designed to manage 100,000 to 1 million patients. And I just want you to leave with one thought. How many people are going to go out after this meeting and have a dinner? Okay, if you have a dinner, let's say, I know not all of you do it, but some of you will have, say, an expensive dinner and spend $100. If you spend $100 on dinner, at least the way money works for almost everyone is you can't spend that $100 on anything else. That $100 is a resource that spent once is not available. I just want you to think about data differently. Your digital data is a resource that could be multiple purpose, used multiple ways, and has zero marginal cost for reuse. So as long as there's secure, compliant structures to work with your data, you can gift your data multiple times for multiple projects to improve care. And so, one of the things we're building into the next generation of our commons over the next few years is the ability for someone to download their data from someone who has that data, and in a secure, compliant way with informed consent, upload the data to a commons, because the marginal cost of sharing your digital data, and to improve cancer, we not only need tumor sequences from people with cancer, but normals. That's how the Cancer Genome Atlas works. You can contribute data, and that's a gift that you can keep giving multiple times. And if you want to find out more about any of our projects, you could go to our websites. And if you want to actually play with this, you could see us at the end in that corner. I guess we're going to replace the flowers. So thank you.